es el premio María Teresa Miras Portugal al mejor artículo de Jóvenes de la Sociedad Española de Bioquímica y Biología Molecular. No tengo ninguna duda que si hay un premio que pudiera tener el nombre de María Teresa, estoy seguro que a ella le gustaría que fuera este. No conozco una persona que ha apoyado más a los jóvenes en nuestra sociedad que María Teresa, y María Teresa pues la perdimos hace un año aproximadamente. La echamos de menos en estos congresos porque era una persona muy activa, y la Universidad Complutense, que ha estado siempre muy orgullosa de tenerla entre sus profesores, pues tiene a bien de conceder también y patrocinar este premio. Este año lo ha ganado Julia Ramírez Moya, del Instituto de Investigaciones Biomédicas Alberto Sols, de la Universidad Autónoma de Madrid. Entonces voy a invitar al vicerrector de la Universidad Complutense de Madrid de Relaciones Institucionales, Juan Carlos Doadrio.
Bueno, buenos días, señoras, señores congresistas. Me alegra mucho acompañarlos esta mañana en representación de la Universidad Complutense, participando en esta entrega de premios que constituye también un acto de homenaje a nuestra querida compañera María Teresa Miras Portugal. María Teresa Miras es una de las figuras más importantes de la investigación científica en España. De ahí que sea obligado empezar mis palabras felicitando a los organizadores de esta entrega de premios, que son la Sociedad Española de Bioquímica y Biología Molecular, por la convocatoria de unos galardones que llevan el nombre de esta gran científica. Porque es necesario que María Teresa siga presente en nuestro tiempo y demos a conocer su vida y su obra a las nuevas generaciones. María Teresa contribuyó significativamente al desarrollo global en un ámbito esencial en la bioquímica, como es la de los nucleótidos, a través del conocimiento de sus receptores y de las vías de señalización en el funcionamiento neural y en las enfermedades, sobre todo las de carácter degenerativo. Con sus investigaciones llegó a algo tan importante en la bioquímica moderna como son sus propuestas de biomarcadores y de dianas terapéuticas. Ella fue alumna, doctora y profesora durante muchos años en nuestra universidad, llegando a ser catedrática emérita de bioquímica y presidenta honorífica de la Real Academia Nacional de Farmacia. Personalmente, muy amiga de toda la familia, mi hermano Antonio es ahora el presidente de la Real Academia, sustituyéndola a ella, que bueno, acabó no por cese, sino porque ya no podía continuar más. Mi hermano Ignacio, que es el único que no es farmacéutico, era su amigo, más amigo que nosotros que somos los farmacéuticos. Mi padre le dio clases, o sea, que realmente teníamos mucha relación personal con ella. Fue, por tanto, además de amiga, una complutense más, de la que todos sus compañeros y admiradores nos sentimos especialmente orgullosos. Ella sintió el entusiasmo de la bioquímica en los laboratorios de su centro de estudios, la Facultad de Farmacia, llegando a afirmar en numerosas ocasiones que la vocación científica no nace, sino que se hace en los laboratorios. Y su vocación, convertida posteriormente en pasión, surgió entre las paredes del edificio de su facultad, en nuestro campus de Moncloa, gracias especialmente a sus profesores y también a la inestimable ayuda de sus compañeros durante todos los años de su actividad docente. Porque nada de lo que fue María Teresa Miras se puede entender sin conocer la importancia de su formación, de quiénes fueron sus maestros y, naturalmente, de quiénes fueron sus colegas contemporáneos, en los que acabó siendo la consolidación de la bioquímica española dentro de la vanguardia en la investigación científica mundial. Brillantísima en todas las fases de su carrera, desde su premio extraordinario, el Premio Nacional Fin de Carrera, pasando por los doctorados de España y Francia, para continuar con entrega y brillantez en la formación de decenas de profesionales a quienes dirigió sus tesis, sus trabajos postdoctorales y sus múltiples proyectos científicos realizados, donde ella aparecía como investigadora principal. Gracias, entre otras cosas, a los premios que entregamos hoy, la ausencia física de María Teresa no hará desaparecer ni su obra ni sus descubrimientos científicos. Su ejemplo permanecerá siempre presente durante años entre las personas que se dedican y nos dedicamos a la investigación. Entregamos hoy los premios que llevan su nombre, en los que reconocemos los mejores artículos científicos publicados durante el pasado año académico. Esta entrega, además de ser un acto público de reconocimiento a los dos premiados, la doctora Julia Ramírez Moya y el doctor José María Santos Pereira, es también un acto de reconocimiento a sus maestros y a sus familias, porque sin ellos los hoy homenajeados seguro que no hubieran podido alcanzar los objetivos de excelencia que hoy celebramos. Por eso, la Universidad Complutense quiere estar a vuestro lado en este día en el que os agradecemos el gran mérito y el esfuerzo realizado en vuestros trabajos de investigación. Ser los mejores, además de una satisfacción, os exige también una mayor responsabilidad para con los demás. Con los demás, y acabando, muchas felicidades a cada uno de vosotros y enhorabuena también a los colaboradores y compañeros de equipo con los que habéis trabajado, reiterando mis parabienes a la Sociedad Española de Bioquímica y Biología Molecular por organizar este acto tan emotivo y entregar estos premios UCM, María Teresa, Miras, tan importantes que nos han convocado el día de hoy. Les agradezco el que hayan escuchado estas palabras.
Iba a recogerle en nombre de Julia Carmen Mayoral. Pues ven aquí. Sí. Sí que hace falta. Entonces está aquí tu compañera. Salúdala. Y quiero llamar también a José María Santos, que viene por aquí por la escalera, que va a recoger la tesis de este premio. Gracias, Julia. Y el título de la charla que nos va a dar es an ADAR1-dependent RNA editing event in the cyclin-dependent kinase CDK13 promotes thyroid cancer. Adelante, por favor.
Primero, agradecer por este premio y por la oportunidad de poder compartir aquí mis resultados. Me encantaría poder estar allí en persona, pero como acaban de comentar, pues estoy haciendo aquí mi postdoctorado en Boston. Pero bueno, si algo nos ha dado estos últimos años es la posibilidad de Zoom y de haber aprendido sobre ello y puedo estar allí de cierta manera. Well, so today I'm gonna talk about what was part of my PhD project and that we published last year in Molecular Cancer. And the title of this publication is an ADAR-dependent RNA editing event in the cyclin-dependent kinase CDK13 promotes thyroid cancer. So first, I would like to introduce what means. Wait a second. Yeah. Yes, so I would like to introduce RNA editing. So this process is the hydrolytic deamination of the carbon 6 of the adenosines. And what happens is when this ADAR enzyme, that is the one that catalyzes this reaction, deaminates this adenosine, we got an anosine. This anosine has a really similar structure than guanosine. So all the breeding machinery of the cell and all the components of the cells are going to recognize this anosine, same thing as a guanosine. So we could say that A2I editing has a really similar effect than an A2G substitution that could happen in the DNA, but in this case, it's happening on the RNA. So it could be cell-specific, time-specific, and also dynamic. So the enzyme that is in charge of this catalysis is ADAR1, that it's ubiquitously expressed in all the cells of our body, and it could edit different parts of an RNA. So the first thing we would think about is that it could edit the coding region of an mRNA. So if it happens in this coding region, we could have a protein with a different amino acid from the same mRNA. So this could have obvious implication on the final function of the protein. Also, it could happen in an intron, which could make changes in the mRNA stability, but also if it happens in what we call cis elements of splicing, that is, these nucleotides that are important for the recruitment of splicing elements, it would change the alternative splicing of the mRNA. So we would have different isoforms of this RNA. Also, it could happen in the 3' UTR of mRNA. This could also change the mRNA stability, but it also could change the regulation of this mRNA. So, for example, it could form new target sites for new microRNAs, or it could avoid deregulation of sub-microRNAs that were regulating this 3' UTR before. And finally, it could also happens in the microRNA itself. So this could disrupt completely the maturation of the microRNA, but also it could happen what we call retargeting. That it's when this editing happens in the seed region of a microRNA, this microRNA is gonna start recognizing different targets than it was doing before. And in the opposite, it's not gonna recognize some targets that it was doing before. So what was really interesting for us is that some groups had studied the TCGA data, or the Cancer Genome Atlas data, and they noticed that there's some tumors in which there's an under edition, so there's a lower activity of editing of these enzymes, but in most of them, there's a over editing. The tumors in which we find a stronger signal of over editing, it's head and neck, breast cancer, thyroid cancer, and lung cancer. The laboratory in which I was doing my PhD, that is the laboratory of Pilar Santisteban, we were really interested in the thyroid gland and thyroid cancer. So it was really interesting for us that this over editing occurs in this cancer, and there was not any other study that studied this process in this cancer. So we thought it could be a good model for that. Thyroid cancer, it is the most common endocrine malignancy, and it's really surprising that the new cases and the incidence has really increased in the last few decades. Usually, thyroid cancer has a really good prognostic because it has a treatment that is completely remove the thyroid and then give to the patient radioactive iodine that it's gonna remove and eliminate all the cells that had a thyroid origin. However, that some subtypes of thyroid cancer that are resistant to this therapy and had a really, really bad prognostic and around 6 months of life for these patients after the diagnosis. So the research for new therapeutic targets and for new treatments for this type of patients, it's really important and encouraging in this field. So what we want to know is what's the role of ADAR1 in cancer. If it looks like ADAR it's overactivated in this cancer, we want to know if this has an effect in the cancer progress, and then if this ADAR is acting as an oncogene in this cancer. If it is acting as an oncogene, we want to know if ADAR1 could be a therapeutic target. And finally, we want to know how is this ADAR1 acting and what's the mechanism of action. So to answer our first question and to wonder if ADAR1 is acting as an oncogene, what we've done is to silence ADAR1 in 3 thyroid cancer cell lines at ascites Cal62, TPC1, and 8505. We observed that we silence ADAR1 with these 2 siRNAs. We observe a decrease in the proliferation of the cells and the cell viability, as we show by MTT and colony formation. But also in the migration process, as we do by wound healing assay, in which we do a scratch or a wound with a pipette tip, and we analyze the speed of the migration, and we see that the cells that are silenced for ADAR1 are really impaired to migrate. And finally, same thing with invasion. For that, we use some transwells that had madrigel, and we see the cells on the top of this madrigel, and we analyze how many cells are able to invade the madrigel. That would be these ones that we have in the picture. And again, ADAR silencing decreased this invasion. We also did some assays in 3 dimensions or spheres formation. It is more similar that would happen in a real tumor. So for that, we grow the cells in madrigel, and after 14 days, we analyze the spheres that are formating. And we observe that in the control situation, they have these huge spheres that even start to invade the madrigel and that even got attached into the bottom of the plate. However, when we silence ADAR1, we have these smaller spheres that always remain as a perfect sphere, and they never invade this madrigel. Because of that, we decided to go to in vivo experiments. So for that, we used again the Cas62 thyroid cancer cell line that were or not silenced for ADAR1, and we inject that into the flank of the mice, and we observed that when we silence ADAR1, we have a decrease in tumor growth. So with all the experiments and some other gain of function experiments that I'm not showing here, we conclude that ADAR1 is acting as an oncogene in thyroid cancer. So then our next question is if we can use that as a therapeutic target. So to test that, we use the small molecule 8-adenosine that is shown in another tissues that it decrease or inhibit the A2I RNA editing by binding to ADAR1. We treat our thyroid cancer cells, Cas62 and TPC1, with different doses of this 8-adenosine, and we observe that it's decreasing the A2I RNA editing by observing some of the really well-known RNA editing sites in the cells, and we observe that this is in a dose-dependent manner. So it looks like it's also working in our thyroid model. Then we analyze the same cancer hallmark that we were looking before, and we observe that 8-adenosine is decreasing proliferation, migration, invasion, and also 3D growth in a dose-dependent manner, concluding that this molecule is able to decrease RNA editing and inhibit the K-cancer hallmarks, so it could be a potential interesting future treatment for this type of cancer. And finally, in a more molecular point of view, we want to know what's the mechanism of action of this ADAR1. So for that, we decided to do RNA sequencing in our conditions that would be, again, this thyroid cancer Cas62 cell line with our siRNA control, and then compare with the 2 siRNAs that we had. After that, we analyzed the differential expression that we got, the editing analysis comparing both conditions, and the splicing analysis. We got some candidates, and then we validate one of them that I told you in the title that is going to be CDK13, describing a new mechanism of action for ADAR1. So about the differential expression, we got 491 genes that were downregulated. Of course, ADAR1 was among them, and 355 genes that were upregulated. When we analyze the function of these genes, we observed that among the downregulated genes, they're enriched in functions like cell cycle, DNA replication, cell division, cytoskeleton mitosis, what really fits and confirms what we were showing in our phenotype in vitro. Also, among the upregulated genes, we saw an enrichment in genes implicated in apoptosis, but also pairs with our phenotype that I didn't show you today, but we also have some phenotype in cell death. Also, it was really interesting that the alternative splicing was a function that was also really enriched among the downregulated genes. And that's why we decided to study the alternative splicing in our comparing our conditions. And we found that we observed 144 genes that present a differential alternative splicing when we compare the SI control and the SI ADAR. And this was observed also in other tissues when they impaired RNA editing. So it looks like RNA editing and splicing has a really relation. We observed that most of the alternative splicing types are represented in our model, although the exon skipping is the one that is most common. When we analyze the functions of these genes that suffer this differential alternative splicing, we observe that the function that is mainly enriched is the alternative splicing itself, what could really explain why we have big changes in the alternative splicing, because the genes that are alternative spliced are also implicated in the alternative splicing. We also observed that we have an enrichment in cytoskeleton proteins, which also fits really well with what we saw previously in vitro. Finally, and most importantly, we analyzed the RNA editing. We observed that we had 122 A2I editing sites, what means that in our sequencing, we see changes A2G, that is how we can show it in our sequencing process, and that this is downregulated when we silence ADAR1, so it's dependent of ADAR1, and of course it's significative, so it has an FDR smaller than 0.05. Surprisingly, there's an overlap between the alternative splicing and the RNA editing. That means that the genes that are getting edited are not the ones that had this differential alternative splicing. So probably ADAR1 is changing a splicing, not by editing these cis elements of splicing, these nucleotides that are important in the mRNA itself for splicing. So probably it's affecting trans elements. That means these genes that these proteins that are implicated in the splicing itself. When we analyze where are these editing sites, we observe that most of them are in the free prime UTR of our RNAs, but we had one missense editing, what means that we're changing the amino acid of the resultant protein. So we decided to study this one because of the obvious function that it could have when it changes an amino acid of a protein. It's like mutating this protein. And we observed that this is happening in CDK13, the position 103, that is in the N-terminus of this protein. And of course, it was significative in our study. What do we know about CDK13? Not a lot. It's a gene that we know a little bit about its function. There's some studies in cancer, but it's still in early development. And there was not any study in thyroid cancer. So what we know is that it is a cyclin-dependent serine threonine protein kinase, and that it has these RS domains, what makes this protein a serine arginine protein family, and that this family is required for splicing. Some of the studies that had studied CDK13 before said that it has a function in the splicing and also in the transcription. So as we said, that probably ADAR is editing these trans elements that are implicated in splicing. We were wondering if this editing CDK13 could explain the effect of ADAR in the global splicing patterns when we silence it. So first, what we've done is to validate the CDK13 editing. So for that, we did a PCR PCR of the region, and then we Sanger sequence it. We observed that in control cells, in this case in Cal62 thyroid cancer cell line, we see that we have this red peak, that it would be the wild type form, and then we have this blue peak that is the edited form. So we have both forms in the control conditions. However, when we silence ADAR1, we just have this red peak that is the wild type one with both siRNAs. So this editing site is ADAR dependent. And finally, we just have the wild type form in the gDNA, what confirms that this is not a mutation, this is an RNA editing site. Same thing happened in the other cell line that we were studying, TPC1, in which we have even more edited form than wild type form. Then we want to know, this is a real thing that it's also happening in patients. So for that, we took some tissue of thyroid tumor and the contralateral normal tissue of the patient. And we extract the RNA, RT, PCR, and again, Sanger sequencing and quantificate the peaks. And we observe that we have an over-editing of CDK13 also in these tumors compared with the normal sample. So this is happening also in the tumor of the patients. It is known that CDK13 is over-edited in other tumor types. When we look at the supplementary material of these huge studies that studied the RNA editing in a lot and a huge number of tumors, we observed that CDK17 appears to be edited in hepatocellular carcinoma, glioblastoma, and also in kidney, renal cell, clear cell carcinoma. And we were also working in breast cancer cell lines because we had some collaborators that were interested in RNA editing also in breast cancer. And we observed that by Sanger sequencing, we see that in these two breast cancer cell lines that we were studying, we observed also this double peak. So it's also happening in these cell lines and known when we silence ADAR1 and also know in the gDNA. So it looks like, and we believe that this CDK13 editing, it's important for thyroid cancer, but it's probably also important in other cancers, and it's also appear in these tumors. So great, this is happening in thyroid cancer, but now we want to know what's the advantage of this editing. So for that, we overexpress the wild type and the edited form, linked to an HA tab because we don't really have a good antibody for CDK13 at the moment. And we analyze what's happened with the phenotype of our cells. We observed that when we overexpress the wild type form CDK13, we have an increase in proliferation. So it looks like CDK13 is acting as an oncogene also in this cancer. But however, when we overexpress the edited form, we have a higher proliferation that is bigger than when we overexpress the wild type form, and we are overexpressing it in a similar amount. Same happened with invasion. The wild type form increased invasion, but the edited form increased even more this invasion. So it's promoting a higher aggressive phenotype in these thyroid cancer cell lines. Then we want to know if this editing in CDK13 has a real implication in the phenotype of that confers ADAR1. So for that, we silence ADAR1 and we rescue the amount of levels of CDK13, but overexpressing the edited form. So we observed that when we silence ADAR1, we have a decrease in proliferation, as we saw before, but when we rescue the levels of CDK13 together with the silencing of ADAR1 that would be here, we rescue the proliferation in a similar way to the control. Same thing in invasion, concluding that the editing of CDK13 rescue the proliferation and invasion defects that we observed in the ADAR1 silencing cells.
Julia.
Okay, perfect. And finally, we want to know what implications this editing has in in the function of CDK13. So there's a group that demonstrated that CDK13 shuttle between the nucleoplasm and the nuclear speckles, and that this is dependent on the N-terminal region of CDK13. And we know that our editing is happening in this N-terminal region. Importantly, the nuclear speckles are some nuclear domains that are enriched in pre-mRNA splicing factors. So probably if we're changing the localization from these nuclear speckles, we're also affecting the splicing function that CDK13 has. So to know that, we analyze the localization of CDK13, and we stain the nuclear speckles by staining SC35. And we observe that in the wild-type form, we have CDK13 in the nucleus, but it's more concentrated with these bright dots that completely correlates with SC35. So CDK13 wild-type, it's mainly in these nuclear speckles. However, the edited form is not concentrated in these nuclear speckles, but is all over the nucleoplasm. So we have a lower colocalization with these nuclear speckles. Also, when we observed this pattern on the wild-type form, we see that we don't have CDK13 in some structures that look like the nucleolus. So we decided to stain the nucleolus with fibrillarium. We observed that there's no colocalization at all between CDK13 wild-type and the nucleolus. However, in the case of the edited form, is all over the nucleus, so it's also in this nucleolus. So it has a higher colocalization with this fibrillarium. Surprisingly, just in the edited form, there's about 15–20% of the cells that present this huge accumulation of CDK13 that completely colocalizes with the nucleolus. So it seems that this editing is making that CDK13 is not anymore in these nuclear speckles concentrated, and is all over the nucleus, and sometimes in around 15–20% of the cells is really concentrated in the nucleolus. So this would probably have an effect in the splicing function. So to confirm that, we just did some qPCR assessing the events that were deregulated in the splicing events that were deregulated when we silence ADAR1. And we observed that when we overexpress the wild-type form, we don't see any change. But when we overexpress the edited form, there's a downregulation in some of the events that were upregulated when we silence ADAR1. And in the opposite, there's an upregulation in some of the events that were downregulating when we silence ADAR1. So CDK13 editing is inducing some changes in this splicing pattern. So as a conclusion, ADAR1 is editing CDK13. CDK13 is over-edited in thyroid tumor samples. The editing event that we're describing in CDK13 is promoting cancer cell hallmarks, and it's changing the nuclear localization of this protein, and probably explaining why it induced changes in the splicing pattern. So as a big conclusion, we are showing a new mechanism of action for ADAR1 that explains, at least in part, why it has an oncogenic role in thyroid cancer. So I would like, of course, to acknowledge all the laboratory of Pilar Santisteban that helped me a lot with this project, and also our collaborators here in Boston, or Frank's lab, in which I made some summer internships during my PhD, and also collaborate with this project. And of course, the organizers for giving me the opportunity to share my work, and all of you for listening.
Muchas gracias. Gracias, Julia.